Okay, we'll go ahead and get started here. Right on time, 1 o'clock. A few of you are still filtering in. There's a few seats kind of up in the middle here in the front. So you guys showed up for Commodity Classic this year. I took the under on attendance for this session, so now I owe my buddy dinner back there. So thanks for rolling in early before the show got kicked off. My name's Jeff Divan. I'm the Director of Agronomy for Sound Ag. Along with me here today speaking is Kakouridis, research scientist, resident AMF expert, so she'll answer all the tough questions when we get there. So what we're going to do here this afternoon to get started is just talk a little bit about AMF. So Arbuscular Mychorrhizal Fungi, that's the first and only time I'm going to say, and it's AMF from me from here on out. We're going to talk about what they are, how they work, and why you should think about them on your farming operation from an implementation standpoint. So before we get into some of those details, I think it's important to get a little backstory on what led us to the point of adding fungi to a soil that already contained fungi, right? Like how are we at this point of imbalance where we're adding something that should already be there? And I think that's where I'll start before we kind of get into the details of how it specifically works. So if you think about the last 100 years or so, in the US, we've increased our productivity by about 700%. So naturally, we're going to need to supplement fertilizer to grow those kinds of yields and take that kind of output off the field. The issue is we make most of those decisions around fertility, around logistics, and around convenience, right? So we're going to put fertilizer on that field at a time it's actually not needed. Let's think of when corn is going to uptake the majority of its nutrients, second half of the season. When do we put all that fertility there to produce that yield? The spring before that season, maybe even the fall before that season, because it was convenient. And we need to cover acres at certain points in time, but it exposes us to loss. So I'm sure you guys all have some variability in weather, just like myself. I'm from north central Iowa, farm there as well. We have springs that are pretty unpredictable. So if you've got a lot of fertility out on that field, and you know that it's prone to loss from weather conditions, or even prone to tie up from chemical imbalance, what has our strategy been? Put on a little extra, right? If we put on a little more, we can lose a little and we're still going to be good. We really start to tip this teeter-totter one direction, especially at the beginning of the season. So there's obviously going to be environmental cost to doing that and having extra fertility out there, but there's also going to be effect to soil biology. So when soil biology and microbes are swimming in nutrients, right, we do everything we can to apply fertilizer next to the seed, under the seed, above the seed. We just pack it in as close as we can. The plant senses that, right? So why would they put energy into recruiting and interacting with biology if they're already swimming in the nutrient that they want, right? The answer is they don't. So what we end up with are essentially kind of a little bit of a lazy system underground where it's not putting energy into recruiting microbes because it thinks it has what it needs. But that's early in the season when we put all that fertility there, and we're kind of seeing if we can get to the end of the game without running out. So, I like to think about this like right after we've had a really big meal, right? How excited are we to go to the grocery store to buy something for the next meal? Or think about starting to cook dinner, right after we're sitting after a big lunch. We don't. It's not top of mind, right? And biology and plants and microbes and this interaction, it feels the same way. We're swimming in it. We don't need to think about our next meal until we do, which is the end of the season. So what do we do about it? We've kind of established now that we've got this system that's a little bit lazy. It's off to a sluggish start in the system. We hear all about these microbes, right? Billions and billions of microbes. But we don't often talk about if we have the right microbes in place at any given time throughout the season. So this is where we kind of enter the issue of Midwest soils are very bacterially dominated. So after 100 years of tillage, which damage fungi, after 100 years of rotating less and less crops every year, we have a balance that's very heavy bacteria, pretty light on fungi. Bacteria can do good things in the soil, but they're not the magic makers that Ann will kind of point out later here of what fungi and AMF specifically can do for us. So we have this big gap in plant health benefits related to season-long access to water and nutrients, as well as pumping carbon back down into the soil. So bacteria aren't going to add it to the soil like we are with fungi. We could go to no-till, and in some places that's an option. It has been for a while. That's a way to start making progress in the right direction. We could also go enter more crops into our rotation, two things that may or may not be easy to implement depending on where you live, and also probably take a little while to see some of those benefits. So what's the approach here on actually finding a solution that's going to impact our fields, our yields, and ultimately our profitability? And that's a multi-pronged approach, and that's where we tackle this question a little bit differently than others. So first and foremost, you need to add high-quality microbes, right? It doesn't matter if you're talking AMF here or some other kind of microbes. They need to be alive, they need to be high quality, you need to pay attention to how you put them on the field. But what's really important here is what I mentioned to start is what environment are they in? If you add a microbe to an environment that the plant already doesn't care they're there, you're likely not going to see a result. And that's some of the issue that we've had with biologicals in general is they're introduced into an environment where the plant is not trying to form a relationship and associate with them. So the key here in the middle is a product we've been selling for a few years called Source that actually mimics that plant to microbe signal to recruit them to the root and establish that connection. Once the connection's established, they can then feed that plant throughout the season. Plant can then put carbon back into the soil and have that season-long relationship. So that's really the key. The end result here is high-quality microbes in the soil, combine it with the chemistry to activate them, make sure they're actually associating with the plant, and the benefit is really consistent results. So I'm going to dig into those results that we have from the past year after I hand it over to Ann and we kind of deep dive into what AMF are once again, and then just make sure that we are talking about the same thing. Ann, I hand it to you.
Hi, thanks, Jeff. Great, well, let's talk about what are AMF more specifically and their impact. I like to start by showing some images of what they look like so you can kind of picture what this is about. And so AMF are fungi, so they are a group of microbes that persist in soil as spores. The spores basically look like very small balloons that contain lipid and DNA, and those can persist for many years dormant in soil. Once they grow, they will form hyphae, which are really thin strands. And then inside the roots, when they colonize roots, they will form some vesicles and arbuscules. The vesicles are really similar to the spores, except they are inside the roots. The arbuscules are where the exchange is going to happen with the roots, so they have a very high surface area to volume ratio. They basically look like very, very small broccoli. And actually, we're having a mini science center at our booth tomorrow where you can come see these firsthand if you don't want to take my word for it. So come visit us. And how do they work? How do AMF work? So those dormant spores in the soil, once they wake up, they're going to send very long and thin hyphae into the soil. And these hyphae are about 10 times smaller than a strand of hair, and they are translucent, so you're not going to see them with the naked eye. They don't make any structures you can see, but you will see their effects on plants. They will wake up when they get a signal from the plant, and the first signal is actually strigolactone, which our main product source mimics. And that's going to help the spores wake up and then grow towards the roots by following that signal, and then colonize the root. The spores have a small amount of lipid reserve, so they can wake up and grow a little bit. But if you want to actually get a successful fungal network, the spores need to connect to the root so they can get sugars and lipids from the roots. That's the only way that AMF can get food. And that's because they don't have any way of doing decomposition or obtaining any sugar or lipids from the soil. Like everything will come from the plant. They have this obligate relationship. And so there's this dialogue happening with the plant that will help this symbiosis establish. And once that happens, then the AMF can grow even up to a yard away from your root and form a really big network that help the plant occupy much more of the soil volume. And so our product Source helps the spore wake up and colonize the plants. And I like to think of AMF a little bit as introverts. If you are yourselves introverts or have introverted friends, you know that sometimes they need a little bit more of a push if you want them to come out and do things with you. But once you do, they will be very good, loyal friends. And I feel like that's really what AMF are. They are dormant in the soil, and you need to help them wake up, but once they do, they will provide so many benefits for the plants. In our lab experiments, we on average see a 2 to 3-fold increase in AMF colonization when we apply Source as a foliar spray. These numbers can be higher. Sometimes you get 20 times more, but just on average, over many experiments, like 2 to 3 times is what we consistently see. Why are AMF special? I feel like we talk a lot about bacteria and what they do for plants. And bacteria are really important because they are the ones that can fix nitrogen. Only bacteria can do that. They also do decomposition, and they release phosphorus, potassium, many other nutrients. But bacteria can't really move around very much unless they kind of follow water, but they kind of are where they are. What makes AMF really special is that they occupy at the same time multiple spaces. They are inside the root, and they also are inside the soil. So they can carry nutrients from the bacteria, where the bacteria release them, all the way back to the roots. And they actually even help bacteria move around. There are water films on the surface of hyphae, and bacteria use the hyphae kind of as little water slides and to go to different places. And so the AMF, because they're really thin, they're able to go into soil pores where roots can go. Roots are really, really efficient at picking up nutrients, but they are much bigger. They are kind of like a highway. So, many cars are on there. But at some point, if you want to go to different locations, you might need to take a smaller road. And that's what the AMF allow the plants to do when they extend the root system. And so, based on all these features of AMF, how do they benefit the plants? AMF are said to be the most important symbiosis on Earth. I know scientists tend to always say that about their own research, but in this case, it's true. About 80% of plants on Earth have AMF, and that's true for natural systems, like tropical forests have really shallow roots and probably wouldn't exist without AMF, but that's also true for agriculture. And the reason AMF are so important, what they're mainly known for is transporting nutrients to plant. And that's phosphorus, nitrogen, potassium, microbes, like even some rare amino acids. And they're really, really good at that. They also help plants tolerate stress better, especially drought, and they help protect roots against pathogen. And those are benefits you're going to get like as soon as the AMF colonize. You're getting all these benefits throughout the season. In addition to that, AMF also provide long-term benefits because they help, with their high-fall network, they help more organic matter accumulate, they help aggregate form, they help increase water holding capacity. So on the long term, like year after year, they will improve your soils. And they're really important in supporting ecosystem function, like all around the world. So in the lab, I have access to some AMF, and I can add it to my pots in the greenhouse. But what about in your fields? There are different areas based on soil type, based on climate, based on geography, that naturally have less AMF. And so in this case, you may want to add more AMF. There are also some practices, such as tilling or adding fungicide, that sometimes you have to do, but those are going to hurt the AMF and reduce how much AMF are in your soil. And so again, in this case, you may want to add some AMF. And so to do that, we now have a new product called Blueprint that contain a really high concentration of active spores. There are many products on the market, but AMF products are actually pretty tricky to make, and for a product to work, it really needs to have a lot of active spores. I've personally looked at many, many products, and a lot of them just don't really have active spores, like the spores might be dead, or you might need to add 10 or 100 times more for it to work. And so we really wanted to have a product that has a lot of active spores. And so I'm going to show you just some images of what it looks like under the microscope. Going from left to right, it just increases magnification. And so you can see the spores that look like little balloons. And there are also little pieces of roots that, if they were colonized, those can help also as inoculum. So just many, many spores. I personally counted them. In a tiny, tiny drop, there are hundreds of them. And not only are there many spores, but those spores are active. On the right side, you can see some of the spores are yellow and some are purple. Maybe it looks just darker, but you can see there are different colors. And that's an assay that we use to check viability. I think everyone has heard the mitochondria is the powerhouse of the cell. And if the spores have active mitochondria, when we add this yellow dye, it will make the dye purple. And so then we can measure how much purple there is to see how active they are. And so we do see them turning purple. Unfortunately, the purple dye then kills them, so then that assay is done. Separately, we also do a germination assay to make sure that not only the spores are active, but they can also produce hyphae. And so here on the bottom left, you can see a spore that's germinated. It has all these white strands compared to one that's not germinated, like it didn't do it. And so for our product, like we have made sure that it does all these things so that you get all the benefits. And so to summarize, our product Source and Blueprint really work really well together. Source activates the beneficial bacteria that fix nitrogen, solubilize phosphorus, solubilize potassium. Source also activates the Blueprint spores to wake up and colonize roots. In turn, Blueprint spores then help pick up all those nutrients that have been released by bacteria and bring it back to the roots. So together, they really are making a powerful synergy that help plants access more nutrients and water. Thanks. Back to you, Jeff.
Yes. All right, so now we've kind of got the setup of why this can be an issue, and then just a reminder on what AMF are, how they work. So now kind of get into what is the result, right? So it's this equation, 1 + 1 = 3, right? That might be what the cash flow you gave your banker this year looks like at the end of the day. But we don't often see that with product synergies, right? Is you can't make something from nothing. And we just see that here for kind of a unique reason, I think. Like I said, not just providing the primer, but actually providing the mechanism for action with these two products together. So this is what we found out this year. Just kind of saying this 1-2 punch of biology plus a chemistry. We got 32 strip trials this year completed with the stack of these two products. So when I say strip trials, I'm not talking about a couple 100-foot plot. This is anywhere from an 1/8 of a mile to a full mile passes across the field, multiple environments in a commercial setting with the commercial equipment. So as real as it gets. And what we saw were results, 7 to 12 bushels. What probably excites me most about this is the win rate at 85%. So this is just something you don't typically see when you're working with natural biological products, because you don't always align environment with the opportunity quite perfectly. And that's what we're substituting Source to do here in most cases. So it was really great to see that come in from really across a lot of the corn producing states this year. So kind of moving beyond yield for just a second to, where did the yield come from? It's got to come from somewhere, and typically we'll see that in plant physiology. This is just some examples from this year. So you hear oftentimes, you don't want your corn to have a bad day, right? But what does that mean? When a corn plant is experiencing stress or abiotic stress, it'll actually just go into a defensive mode and it'll stop growing. So it's trying to conserve all water to stay alive, and it essentially stops in its place until the weather maybe starts to calm down or you get a rain event, something to that effect. So the result is a plant that was not experiencing that stress is going to continue on its growth curve. And you see that especially in this picture here on the right, where the first plant is popping a tassel, the one next to it is not. That's a difference of about 2 leaf collars. The only difference in that field was AMF and Source to activate those AMF. So they didn't experience as many bad days. When you look at modern current hybrids, where they really pack on yield is in grain fill. The quicker you can get that plant to grain fill and the longer you can keep it there, the deeper you're going to get those kernels, the more you're going to drive yield, right? So we got it there about a week quicker in this standpoint. We got slightly longer days in that week, and we kept it there longer with some crop protection products longer into the season. And that's where you get results from a yield perspective. So just wanted to show that. That's kind of some of that where you don't want your corn crop to have a bad day. You don't want it sitting there not growing. That's costing you bushels on the tail end of the season. Okay, so to get back into the yield data, it's just an interesting cut here. A lot of times we'll validate products and we'll look at entire strips and blocks across the field. So what was the average of this one? What was the average of this one? We worked with a partner here to parse out one of these trials a little bit differently and say, how did they compare based on slope of that field? So not just reacting block for block, but what did the 1% slopes compare to the other 1% slopes and so on? What you'll see here, not much impact on the left side of this chart. So if you can't see it, just the control is the light green, the Source and Blueprint is the dark green. So as we work from flat, perfectly flat on the left side to right, you'll see those bars start to separate. So we go from almost no impact in the perfectly flat black part of that field to a 25 bushel yield lift on the 4 and 5% slopes in the field. Why is that? a slope in a field is going to do a couple things. Number one, it's going to shed water faster than any other part of the field, right? Water moving away from part of the field is going to affect a couple things. It's going to affect nutrient uptake. It's also going to affect how quickly that part of the field gets to a point of experiencing abiotic stress, like I just mentioned. So if you think what Ann just showed, better access to water outside of that depletion zone of the rhizosphere within an eighth of the root. So we're going to get water beyond an eighth inch of any root. We're also going to be able to interact with bacteria to pull more nitrogen, pull more phosphorus. So we now went from a field that was, part of that field that was stressed to a pretty extreme degree in this case, and brought it up yield-wise to exactly pretty much the same as the flat part of that field. So if you look at the dark green lines, what's very interesting is you removed almost all variability from that field. All right, it's only about a 5 bushel variance in that 18-acre block, and all it was coming from, or primarily what it was coming from there, was slope and water leaving parts of the field. So now with AMF active and associated with the corn plant, we're able to mitigate that and drive yield, especially in those parts of the field that were challenged. So lots of opportunity there long-term to say, let's invest in that slope part of the field rather than just write it off and write a lower prescription. Okay, so to go below ground a little bit in results, lots of different ways to measure soil health, right? If we asked you guys how to measure soil health, probably get 100 different answers in this room. We chose the Haney test. It's been around for a while. It's got a lot of different components that we think are pretty compelling. So we went out in those trials and we did a protocol where we tested within the treatment, and a very adjacent spot we tested untreated. And here's a few things of what we found was, number one, CO2 burst, which is on the left. That's going to be a soil respiration measurement and basically tell us what is the overall biological activity in that soil sample. So we can see we increased it by about a third in this example. Next column in the middle is the aggregate of the soil health score in general. So that's looking at a combination of respiration, water extractable carbon, as well as nitrogen. So kind of putting all three of the building blocks together. Once again, we saw an increase of about 17% in this example. And then the furthest right bar there is the microbial active carbon. So it's the energy available to microbes. And if you listen to what Ann said closely, not only are AMF pulling in water and nutrients, they're also pushing back out and moving the other direction with a food source for biology. You have to feed biology in order to benefit from it, fixing nutrition. So that's just a few of the outcomes of the Haney test that we looked at this past year. So another way to consider the impact of this system is how many pounds of available nutrients were there in that soil at any given time once they've been impacted by AMF. So pretty simple here. We looked at water extractable N, P, and K, so plant available forms. Nice, about 9% increase in nitrogen. Huge one here, almost 25% increase in plant available phosphorus. There is a ton of phosphorus in our soils. It's just locked up, right? We're either not solubilizing it and it's bound up tightly with a metal, or it's not getting transported to the root. So we're solving both of those here. And then finally K. K is an access issue, right? It doesn't move much. We got to go find it. It doesn't find our plant. So we're benefiting from increased root growth and AMF acting in that way. So that's kind of the below ground portion of why we got those results. So just to kind of summarize here the entirety, and we'll take time for questions here. This worked out good for timing. We have set up a system where we are limiting the impact of AMF in our soils. That's from management practices and what we're doing from a cropping standpoint. That's fine. We have tools where we can remedy that. We can add AMF to the soil. We can activate them with source. And then ultimately at the end of the day, we're going to benefit in the terms of yield. And a consistent yield approach we haven't seen without putting biologicals together with chemistry. So yield, soil health, available nutrients, that's our benefit as producers from putting AMF to work in our fields. So with that, I just want to mention two things. Number one, we have a happy hour in our booth. We're right by the main entrance. I think it's booth 175. So happy hour this afternoon, I think about 4:30. We'll also make you a hat there if you stop by. If you scan that QR code, it's got more information, take you to our website, sound.ag. You can learn more. But with that, we've got about 6, 7 minutes for questions. There's 2 microphones that are going to run around the room. If anybody's got a question, just put your hand up.
Got one back here, Jeff.
So if I'm understanding, is the blueprint part a new addition to the source product, or is it in addition to that would be mixed in separate?
Yeah, it's a great question. So it's a separate product format. So Blueprint is going to go in at planting as an AMF inoculant. So there's two formats. There's a dry powder, there's a liquid. You could either do an over-treatment on seed or in furrow with a liquid system. And then you'll come back and spray Source V4 to activate that symbiosis. Yep, so separate product.
Sorry.
Well, grab the mic. They're recording this, so then then I don't have to say it again. Thank you.
How are we for state labeling on the Blueprint?
Yeah, vast majority of states. I don't have the list in front of me, but anywhere east of here, you're fine. If you're west of here, come to the booth.
So if I understand correctly, the source, if we were looking at small seed crops, is a seed treatment. Did I understand that correct?
Source is going to be a foliar applied. It mimics that signal out from the plant to the microbiome. So it's going to be foliar applied on everything, legumes, cereals, whatever it may be. AMF is going to be applied at planting.
Okay.
Through Blueprint.
And so with that, in an area that's really low in manganese, and we seed prime also right on the seed with a manganese, and manganese is pretty tough on a lot of fungal, is there an interaction that's negative there?
You're good.
I would have to know the exact concentration. Sometimes it could harm them if they're in direct contact, but usually they can, like AMF are pretty resistant to those kinds of things. Yeah.
So what is the life cycle once a spore germinates, and what is its benefit to the plant? Is it only for the season, or a week or a month, or what does that life cycle look like and benefit to the plant?
Great question, thank you. Yeah, so they're going to colonize. That takes a few days. Usually, like they germinate, colonize the plant, then after a few days to a couple weeks, they start transporting nutrients and benefiting the plants, and that's going to continue throughout the entire season. And once you have your big hyphal networks, they are going to actually make more spores that can then persist and colonize again next season. So it's really, if you add the AMF multiple years in a row, that can still be a benefit because you want to really build your population, but with a single application, you will get a benefit for multiple years.
Is all of your guys' research done in conventional tilled situation, or is there a difference between conventional versus no-till?
Yeah, so there's two forms of research. So what's done in the lab, obviously it's hard to recreate tillage scenarios. In the field, we had a variety. I would say that typically you have a higher baseline in a long-term no-till situation than you do in conventional till, but we still saw pretty low levels from a beginning population standpoint in all soils. So I think there's a lot of room to benefit, especially if it had tillage history, maybe before you started on no-till. Lots of room to benefit there, but. Yeah, you can swing by the booth. We can look at some specific fields that have practices listed as well.
Has anybody applied Source to cover crops and stuff like that to activate sooner, to mineralize or get the speeded up or whatever?
Yes, so we've had it applied to cover crops and then attempted to measure efficacy in the following cash crop. So interestingly enough, when you apply Source, especially in I'm going to zoom back for a little bit to longer term pasture and even grass hay type situations, we see biomass increase a lot, very often, because they benefit from the lack of nutrition at depth in the rooting system. Biomass doesn't always translate into crop yield, right? So biomass increase, yes, especially if you're considering grazing on that would be an option. But to just see it come through in yield in your following cash crop, less consistent results there because we've got to convert that biomass back into something plant available. But we've tried it.
A two-part question. What is the shelf life of Blueprint?
Sorry.
And then what is the longevity of the spores once it's been added to a pop-up fertilizer?
So the powder product, the longevity is 2 years at room temperature. The liquid one is 1 year. And those are really because what we can guarantee, it might really be longer. Spores in the soil can persist for many years. Like, so once they are in the soil, as long as they remain covered, they will be there for several years.
Specifically, if it's added to like a 1034-0, though, like once we've put it into the product, is I think what she says.
Yeah, I mean, no different than a lot of stuff. You want to apply it, I would say, within days. And probably make sure that there's an aspect there with where I set this up of excess phosphate fertilizer in the same location you're trying to colonize AMF can be problematic because they're working against each other from the plant standpoint. So just watch out on not too high levels of 1034-0 if you're trying to get the mechanism of AMF working for you season long. But they can be mixed together. They are compatible. But yeah, we can swing by the booth and we got to watch out on rates just a little bit.
I understand Blueprint is a seed applied product and Source is foliar applied.
Correct. Yep.
Can that be mixed with herbicides then?
Yeah, the Source can go in with any herbicide, fungicide, insecticide. Very compatible there.
With fungicide also?
Yes. Yep. And then, yeah, the Blueprint, just touch on it one more time. The dry product would be applied in the planter box. The liquid product could either go in furrow or as an overtreatment on seed. But it, no, it doesn't have to be on seed. I mean, that's like the in furrow portion, just adjacent.
Like Dosatron, is that way around this,
Yeah, you could go Dosatron just for the sake of the use rate. Yeah.
So operationally, if a Source application doesn't work through the vegetative stage, what's the benefit of just Blueprint by itself?
Yeah, it could be, I'm not sure if I understood exactly where that question was going. Either product can work by themselves, right? Blueprint to the tune of probably a couple bushels per acre. Source probably something like 4, 5. So yes, you could use it by itself for a couple bushels per acre, but we're going to get that magic happening when we put them together for expecting to see something in double-digit area. But yeah, you don't have to use Source. It's just the fact that that plant is not going to be putting out a signaling molecule to try to recruit AMF for that symbiosis.
AMF exists naturally and form natural symbiosis with plants. So that's going to happen even on their own. It's just that Source will really boost the signal, so you will get more of the spores waking up. So you get an additional benefit. But they do both work separately.
Yeah, with that, I think we're there. So thank you for your time and attention, and enjoy the rest of the show. Thank you.